Okay, so good morning guys. So I will be giving you this video lecture discussion so that you might have a guide, no, for the previous discussion, which is all about chromosomal aberration. Also included in this video lecture is the next topic, which is all about genetic mutations. Okay, so for the chromosomal aberrations, no, it is actually a part of the cytogenics. This is a genetics that is more or less focusing on the cells, no? And since chromosomes are found in the cells, therefore it's under the field of cytogenics. Okay, so specifically, when you are going to study chromosomes, you are actually practicing karyotyping. Karyotype is a microscopic examination of the chromosomes. The purpose of your karyotype or karyotyping is to identify and classify chromosomes based on their size, the locations of their centromeres, and their banding patterns. Okay? So basically, this is what is karyotyping. No? You have the chromosomes, okay, the diagrams of the chromosomes, the image of the chromosomes, no? Then you can actually see the specific bandings. These bandings actually is the site of the alleles wherein they hold, no, genetic informations and genetic instructions. Okay. So for the chromosomal aberrations, we have actually three types of chromosomal aberration. First is the substantial change in the chromosome structures, or basically saying the change in the structure in the chromosome, whether it has been cut, it has been duplicated, some parts of it may have been inversed, no, or one part of the chromosome has been translocated to another part of another chromosome. Okay, then we have the typically affect multiple genes. So we have different genes that has been affected by sudden change, no. It could be that they are reduplicated in one or more chromosomes, no. And we have the error in cell division, wherein the cell actually failed not to equally separate the sister chromatids during the meiosis process. Okay, this usually happens to a female that is more or less having a very old age na, and it happens na she is pregnant. So being an elderly woman and being pregnant, no, actually put risk to the child because they are more highly susceptible for non-disjunctions, no, during meiotic process. Okay. Then this is a diagram of how non-disjunction may happen during meiotic division. So basically, we already know that for the non-disjunction, for the meiosis to happen, no, it undergoes two phases. First is the first meiotic division, and second is the second meiotic division. Here on the left side, we have the first example of how non-disjunction may happen. So it could occur first in the meiotic division, and even though that it will follow a normal disjunction in the second meiotic division, all of the offsprings of that particular cell will become affected because of the non-disjunction. On the other hand, we have the second example wherein the disjunction at first meiotic division is normal. Okay. However, for the second meiotic division, the second cell is actually experiencing non-disjunction. So meaning, the offspring of this particular cell is affected with either aneuploid, no, na mga problems. Okay. So actually, there are two types of chromosomal abnormalities. We have the numerical abnormalities, wherein the total number of the chromosomes is abnormal. So it could be aneuploidy or polyploidy. When you say aneuploidy, the number of the set, no, is being added with one or two or more chromosomes, no. While for the polyploidy, it's no longer just a chromosome that has been added. It is an entire set that has been added to the normal size of the chromosome number. Okay? For the structural abnormalities naman, so basically, there is no problem with the numerical number of the chromosome set. Okay, so they are all normal. It's just that a particular chromosome, may it be one or more, has been undergoing deletion, duplication, inversion, or translocation. Meaning, the part of the chromosome has been deleted, a part of the chromosome has been reduplicated, a part of the chromosome has been inversed, no? And some parts of it has been translocated from one chromosome to another. Okay? So, for numerical abnormalities, we have aneuploidy and we have polyploidy. So, for aneuploidy, a normal chromosome set has been added with one or two or more, no, chromosomes. Or, it could be, it is been deleted, no, na-delete na one or two or more chromosomes. Okay? So, for hyperploidy, okay, so this is a type of aneuploidy wherein there is a gain in the chromosome. Okay? So, example of which are your trisomy, double trisomy, and tetrasomy. For trisomy, so it is a chromosome set, a normal chromosome set, added with one chromosome. Okay? For double trisomy, it is a normal chromosome set added with one chromosome to one chromosome region, and another one chromosome has been added to another chromosome region which is different. Okay? For tetrasomy naman, it is a normal chromosome set. However, it is different from the double trisomy because the two chromosomes that has been added is added to a specific chromosome region. Instead of being separated from two different chromosome regions, kani siya is added to the same chromosome region. And ang na-add are two chromosomes. Okay? For the hypoploidy, there is a loss of the chromosome. So this is actually the opposite of hyperploidy. Okay? Monosomy is the opposite of trisomy. So meaning, from a normal DNA set, chromosome set, there is one missing na chromosome. Okay? For double monosomy, so from a normal chromosome set, which is 2N, one is being taken from one chromosomal region and another one chromosome has been taken to another from another na chromosome na region. These chromosome regions are different. Okay? But for the nullosomy, no? So one chromosome region has been actually been deleted. Okay? So there are two, di ba? Since chromosome 1 to 23 are paired chromosomes, so in this case, na-adjoy usa ka chromosome region na na-delete. Okay? So that is for the nullosomy. For polyploidy, actually, it is being added, it is adding an entire set from the normal set of chromosome. Okay? If one set is added to a normal set, then that is a triploid. If two sets are added from a normal set, that is a tetraploid. If three sets is added to a normal set, and that is a pentaploid. And so on and so forth. So here we have a diagrammatic representation of what is a polyploid and a aneuploid. You know? On the left side, you can actually see that from the normal DNA setting or DNA set, no? So it is 2N. As you can see here, each chromosome region is receiving one extra na chromosome, and therefore it is one set. Okay? So from the 2N and plus one set, so it become a triploid. And from the next example, duha ka set ang na sobra, so that therefore it creates a tetraploid. For the aneuploid, as you can see here, you know, the chromosome region number 2 is being added with one chromosome, and you can never find another additional chromosome on the other regions. Okay, so therefore it is a it is an example of aneuploid, specifically a trisomy, no? And it is specific in chromosome 2. Okay, So the number here denotes where the chromosome is added. Okay. Now for this one, we have monosomy 1. So we have the idea that there is a missing chromosome at chromosome number 1. Okay. Now let's move on to the examples of your aneuploidy. Okay. But first, we will be talking all about aneuploidy that happens in a sex chromosome. Okay. So first, we have the Klinefelter syndrome. Okay, wherein there is an extra sex chromosome. Okay, so instead of having 46, it has 47 chromosomes all in all. Okay, so aneuploidy occurs in 1 to 1,000 infants. Okay, they are sterile, meaning they cannot reproduce by the time that they become adult. No, so basically, the features of your Klinefelter syndrome are the same with a normal person. No? It's just that sa mga body niya makakita taog difference. Okay? So they have narrower shoulders and wider hips. So these males actually look like a female. No? So for those person that is actually having Klinefelter syndrome, so they have smaller testes and penis compared to the average male. They have decreased sexual interest, a weaker bone, and a lower energy. And the most prominent trait is that if you are a male, no, supposedly you don't have a developed breast. No? However, for those who is diagnosed with Klinefelter syndrome, they have a developed breast growth. Okay? And they have less facial hair compared to your average male, and they have reduced muscle tone. Next in line is your Jacob syndrome. Okay? So Jacob syndrome, instead of having XXY, this time it has XYY. So it has an extra Y chromosome, and still, instead of having 46, it has 47 chromosomes. So basically, Jacob syndromes are brought about by excessive high level of testosterone during pregnancy, because the mother could be experiencing stress, or the mother is experiencing trauma due to beating of the husbands during pregnancy. Okay? So, Jacobs syndrome is characterized like a normal lanjud na person. So, if you look at a person, you can actually cannot see any difference from a normal person to a person having Jacobson syndrome. However, prominent characteristics could include they have acne, and they are taller than compared to an average male. No? However, the most striking feature of Jacobson syndrome is that people which are diagnosed with this type of syndrome tends to have antisocial behaviors. No? And most of which, the antisocial, they are tightly linked with antisocial crimes, no? Because of their behaviors. No? So, okay. So, it occurs in 1 over 1,000 infants, no? They are basically just lanky, clumsy, and uncoordinated individuals. Okay, they have problems with their motor coordination. They could also have dyslexia. And they have behavioral problems such as behavioral tantrums. They are more impulsive than a regular adult. And they are emotionally immature. And this particular behavioral problems lead them to commit sometimes mga antisocial crimes. Okay? Then we have the Turner syndrome, by which instead of having 46, they only have 45. So allow me to correct this number. That is 45 because the sex chromosome here is lacking a pair. No, it's pair. Okay? So basically, those people that is having Turner syndrome demonstrate visuospatial deficiencies, and they are poor in handwriting, and they have underdeveloped arithmetic skills. Okay, so another features of those who have Turner syndrome is that these are females which are very short for their stature, for their age. They have ovarian failure, they have otitis media, and they have dyslexia or reading disorder. Okay, now enough with the aneuploidy that can be found in the sex chromosome. Then we will be moving with the aneuploidy that can be found in your autosomes, no? So, first in line is your Patau syndrome, which is trisomy 13. Okay? So, trisomy 13 meaning there is an extra chromosome found in your chromosome 13. Okay? So, those individuals that are diagnosed with Patau syndrome have severe intellectual disability. They have physical abnormalities in the many parts of the body. They have heart defects, brain and spinal cord abnormalities, and there is the occurrence of a cleft lip or a cleft palate. Okay? So, mga pinakatamalihan for the Patau syndrome. Okay, then we have the Edwards syndrome or the trisomy 18. No? So, right now, it is the chromosome 18 that contains an extra chromosome. Okay? So, for the characteristics, so they have intestines protruding outside our body. They have intellectual disabilities. They have delayed development. And feeding and breathing and they have feeding and breathing difficulties. Okay. Aside from that, they have arthrogryposis, no, for Edwards syndrome. Okay. Then we have the commonly known na autosomal chromosomal aberration, which is your Down syndrome or having trisomy 21, wherein they have physical growth delays, severe degree of intellectual disability, a short stature, and a delayed mental and social development. Sometimes they also have impulsive behavior. They could show impulsive behavior, poor judgment, short attention span, and slow learning. Addition to this commonly seen traits are your sleep apnea, dementia, and hypothyroidism. Okay? This is a table, no, of sa summary sa atin ng mga sakit, which is in turn showcasing aneuploidy. Okay? So, effects of nalisomy. So, basically, for animals, by the time that nalisomy na happen sa offspring, the offspring immediately die. But for plants, they can still thrive. No? So, for plants experiencing nalisomy, so they are dwarf, okay? And most of the time, they become sterile, especially for the male. But for the female, they can still be fertile. Okay. And then for the effects of the polyploidy, you already know what is polyploidy, right? For the animals, if you have if so happen na ay polyploidy, the animal or the offspring of that animal will also be dead. No. But for plants, it could promote higher yield. Okay, so the more the chromosome set is added to a plant, the bigger and the stronger it gets. Okay, so for the plants, the benefit of having polyploidy could lead to seedless fruits, seedless flowers, and even it could prevent cross-pollination of transgenic plants. Okay, so for the structural abnormalities, okay, we have 4 common structural abnormalities. We have deletion, duplication, inversion, and translocation. Okay? So, for the deletion, from the word itself, so there are regions, or there is a region in the chromosome that tends to be deleted. No? It could be found in the terminal end, meaning the deletion happened at the terminals of the chromosomes, or it could be interstitial, meaning the deletion happens in the middle of the chromosomes. Okay? So, deletions are basically fatal na type of structural abnormalities in a chromosome because it could delete very important informations, no, for carrying out and manifesting a trait. Okay? So, the next is the examples of mga sakit or syndromes that can be caused due to chromosomal deletion. So we have Cri-du-Chat syndrome, by which they also known as cry of the cat syndrome, no, because when they cry, they sound like a cat. Okay, so they have very high pitched cry, intellectual disability, delayed development, small head size, low birth weight, and weak muscle tone in infants. Okay. Aside from the cradle chat, we have the DiGeorge syndrome. This is actually caused by deleting the long arm of the chromosome 22. So, individuals having DiGeorge syndromes have congenital heart disease. They have cyanosis or blue coloration of the skin. They have learning difficulties and other psychiatric disorders. Aside from the George and the cradle chat syndrome, we have Angelman syndrome. This is the deletion of the chromosome 15. So these individuals have uncontrollable laughters and they have poor to no speech development. Okay. For the duplication, so regions of the chromosomes has been duplicated. Okay. So sometimes duplication could be direct or inverted. When you say direct, the part of the chromosomes, kung unsa yung orientation, yun sa siya pag kopya, is the same, no? Pag reduplicate. Sometimes, duplication could be inverted. It could occur, no, na the region that has been duplicated has been inverted. Okay, so that is inverted duplications. Okay. Then we have inversions wherein a segment of the chromosome has been flipped, no? So nabali siya. Now, it could be pericentric, meaning it involves the P and the Q arm of your chromosomes, or it could be paracentric, which involves only one arm, no? So, it could happen, no, in your Q arm alone, or it could happen in the P arm, no? Na-invert ang mga sequences dito. Okay? Okay, and then we have translocation. Translocation is basically, no, one part or one segment of the chromosome, of one chromosome, is being transferred to another na chromosome. So we have two types of translocation. First is the reciprocal translocation, wherein, no, two chromosomes exchange, no, their segments. For example, chromosome number 4 has been translocated to chromosome 20, while some segments of chromosome 20 has been translocated to chromosome 20. Okay? Then we have the Robertsonian translocation, wherein, no, what happens is the part of the chromosome, no, kini, some of the parts has been lost, okay, during the translocation process. So, for example, this part here will be translocated to this another part of another chromosome, and this one, or these two parts, will be lost during the process of translocation. Okay, so much of that, no, for the chromosomal aberrations, numerical and the structural. So we will proceed with our genetic mutations. Okay, so genetic mutations. Okay, so for the genetic mutations, or aka the point mutations, so this time the mutations that we are looking is actually on the genetic level or in the DNA sequence level. Okay, so when you say point mutation, the gene mutation only happens in one gene, okay, or one specific DNA sequence. For example, no, you have your your DNA sequence, diba? And by virtue of the central dogma, you convert it into mRNA sequence in order to create a polypeptide, which in turn will create the proteins, no? And the process involved is your transcription and translation. Okay, so from the DNA antisense strand, or also known as your DNA 3' to 5' strand, you can create the mRNA and the polypeptide. Okay, so you have the normal gene setting here, okay, and then we will convert it into an mRNA, okay, and those codons found in your mRNA will create the polypeptide that is given on the slide, right? So try to, I don't know, try to familiarize, okay? Now, it now creates your polypeptide made up of your different amino acids, okay? Now, you have here the first mutation type, which is your substitution, okay? So again, you have your normal gene here, you have the codons, and you have the amino acid. And you have here on the right-hand side, the substitution mutation, wherein, as you can see here, instead of having the T or thymine, it has been substituted with A or adenine. Okay? So this is a type of point mutation because there is only a mutation that happens in one codon. Okay? Now, instead of having instead of having instead of having CCA GAG, you have CCA GUG. Okay, so there is now a change, no, in the codon, and therefore, because there is a change in the codon, instead of producing this particular amino acid, you create valine instead. Okay, so that is substitution mutation. There is one na DNA sequence that has been altered. Okay, so for the substitution, it will only affect a single codon. Take note of that. And their effects may not be serious unless they affect an amino acid that is essential for the structure and function of the finished protein. Take note also, guys, there are some substitution ng good, no? Wherein, even though they replace the particular code, no, in the codon, but still the codon that has been newly created still creates the original na amino acid, so that's okay. Okay, so walay ma-happen. What is detrimental to this substitution mutation is that if it changes the type of amino acid that should be created. Okay, for instance, like this one, no, instead of creating this particular amino acid, it creates valine. Okay, so therefore it could be, or it could pose detrimental concerns, no? Okay. So the genetic code is generate. A mutation may have no effect on the phenotype, and changes on the third base of a codon often have no effect. Okay, so this is what we call as your nonsense, no, mutation, meaning wala siya nausap. Okay, sige. So you can see here, even though that there is a change in the chromosome, like for example this one, no, like this example, even though that there is a substitution that happened, still the amino acid that has been created are the same. Okay, so there is no change. It is not detrimental. Okay, and it could be disastrous if, no, a substitution mutation led to the promotion of a stop codon, tanawa, no. So as you can see here, this type of particular mutation or point mutation led to the formation of your kanang amino acid creating now the stop codon. So instead of creating pa cysteine and your glycine, ning stop na noon siya. So it could be detrimental because the backbone or the amino acid is not fully developed or not fully created. Okay. Then we have the inversions, no, wherein there is an exchange, no, where to the pattern. Okay, instead of having CT, no, for example, you have here GGTC TC. No, instead of having TC, it has been CT, no. So that is inversion. So nagkabali ang arrangement. And of course, it could lead to different formation of the amino acid later on. So this could be also detrimental. Okay. Now we have additions. Okay, when you say additions naman, it is not anymore a point mutation. Okay, because for point mutation, wala man nag-usab ang number of your DNA sequence. But for addition and subtraction of DNA sequence, it could actually change how the codons should be read. So therefore, there is a frame shift mutation that could happen. Okay, so it changes actually the entire, no, the entire sequence where the addition happened or where the subtraction or the deletion of the DNA sequence that happened. Okay, for example, you have this particular normal DNA gene, and then you insert, no, G in between here. So actually, if you create an mRNA, the codon will also be having a different reading, no, or different translation because you have added another DNA sequence. Okay, so that is frame shift. Okay, kung wala ra kayo nag-add or the number of the DNA sequence are basically just the same, therefore that is just a point mutation. Okay, so I hope I'm clear. Then we have the deletion. It is actually the total opposite of addition. Instead of adding DNA sequence to the original template, you have deleted something in the DNA sequence. You have deleted it's either 1 or 2 DNA sequence, making the DNA short and therefore changing how the codon should be read. Okay, so in the process, it could actually alter the entire amino acid sequence. Okay, so I think that will be all. Okay, so that will be all for our genetics na topic. Okay, so I hope that you have learned for sa atin na tawag na topic, So basically guys, this is the end of our genetics na klase. Thank you for staying with me for the entire semester, and I hope that you have learned something out of our course, which is genetics. Okay, so thank you guys, and God bless.